the main differences and similarities between the de novo assembly and the whole genome realignment, because you all know now how to do the de novo genome assembly and what is behind it. And now we will learn how to do the genome realignment. So the first two steps, doing actually the sequencing, is the same. So you can do it with short Illumina next generation sequencing data or sequencing. You can do any other things, MinION or PacBio. Of course, you will have better or worse results from one and another aspect, but in basically there is no difference between the sequencing data you are working with. There is maybe one thing I should mention here. For the de novo assembly, you really need a good amount and good quality sequencing. So even for prokaryotic de novo assembly, it's good if you have at least a 30x coverage. So you are sequencing 30 times more nucleotide than the expected genome size. And also, it's better if the base quality is good. So it can happen that, for example, you are sending a sample to sequencing and you want to do de novo assembly, but you get back a little bit crappy data. But actually, maybe you don't have to throw that crappy data out. If you can ask questions, if you are, for example, looking for an MLST or you want to make a phylogenetic tree or put that sample into a phylogenetic tree, or you are looking for specific genes in the sample, you don't really have to rely on the de novo assembly always. And if you have a little bit worse data, if you have a bit lower coverage, then you can try with the genome realignment. Of course, you have to find out what's the best reference genome, but it's usually not that hard. If you know your bug, because you are, for example, culturing selectively to Staphylococcus aureus, or you are culturing selectively to Campi or E. coli, then and you think that it will be an E. coli because it looks like an E. coli when it's a colony, or if you do some other biochemical or mass spec identification and you see that it's an E. coli, then you can use an E. coli reference genome. But of course, there are also several thousand different genomes, E. coli genomes, reference genomes. So that's about the input data. But actually, if you have a good quality, good coverage sequencing data, you can do both of them without any problems. The pre-processing is just completely similar, so you can do the same kind of deduplication, and then you can merge, or it's actually not extremely important to merge your reads before the realignment. And the next step is actually the main difference, because instead of creating a novel whole genome, you are just aligning all the reads to a known whole reference genome. At this point, there is a huge difference in hardware requirement as well. While creating a whole genome, doing the whole genome assembly, even on a small prokaryotic genome, it will take a half hour or an hour. If you do hundreds, it will take much more time, of course. And you need that good amount of CPUs, good amount of memory, because if you remember, when you do a whole genome assembly, you basically have to have all the reads in some way within the memory at the same time, because it has to combine all those reads. It has to find all the overlaps between reads. And in the contrary, if you do a whole genome alignment, it has a way much lower requirement for hardware. So even a human a human genome realignment can be done on a simple laptop, because the only thing you have to put in the memory is the reference genome, which is, in a bacterial genome, we are speaking about a few megabytes only. But even if in a high level eukaryotic genome, a vertebrate or a human genome, it's like a few gigabytes of data that has to be in the memory. And when the realignment starts, it actually just reads in the read pairs one by one. It reads in one pair, try to align, try to find a place, and if it's found, then it outputs the alignment result, and that's all. And then the memory freed up. So this is again something you may have to consider if you don't have the chance to access to a bigger computer, or for example, if you are somewhere in like away from your basic hardware background, then you can do a whole genome realignment easily on your laptop. The main goal is again a bit different. So in as we spoke about last time and before, the de novo, if you do a de novo assembly, you will get back the FASTA file, which represents the genome, and then you are starting looking at features within that genome. You are looking at specific genes of your interest. You are annotating all the genes and so on. In genome realignment, you're always looking at the differences compared to the reference sequence. And finally, what you get back is what the working material, which we will see today and which we saw last time. Then over assembly, you have the FASTA file, and you are making GFFs, for example, if you annotate the genome. You are making alignments with known genes and try to find those genes with BLAST, for example. So you are working mostly with a FASTA file or gene annotation files, while in the genome realignment, your primary result, your primary end material is an alignment file, which is actually showing you how and where your short reads are aligning to the reference genome. And then usually the next step is that you are looking at the differences, not by your eye. We will looking at the differences today with BEMView, which is a graphical program to see whole genome realignments, but also you can use different kind of programs to do the variation calling, which will end up with some kind of usually a VCF variation caller file, which actually contains one difference per line, the difference type, the difference position, and what is actually the difference. It's just a SNP, or is it an insertion deletion? So the two approaches are a bit different, and especially the two end products are very different. But also we can sometimes using them to complement each other. So I only put here two examples, but I think there are so many actually. There are quite a lot of other examples. The first example is what the software Ariba does. We are using that program quite often. So if you have a collection of your gene of interest, you can find those genes before you do any kind of assembly or alignment. So you don't really have to assemble the whole genome. If you just want to check that those specific genes are in or not in your sequencing raw data, so this is working with FASTQ data, then you can use the Ariba. And the first step of what Ariba does is that it just makes a simple realignment. I think it is using Bowtie 2 or Bowtie, the program that we will use today, and just try to find those reads that are aligning to your genes. And as soon as it finds something, it will try to extend that sequence. So of course you are just giving the sequence of the gene, which is, let's say, 1000 base pairs from the beginning to the end. But ARIBA also tries to go beyond these 1000 base pairs, 5' and 3' way as well, and looking for those reads that are those short reads that are extending your contig or your gene. The reason behind doing this is because then you can also see what is the sequence environment around your gene. Of course, in many of our cases, when, for example, we are looking for antimicrobial resistance genes that are quite often in plasmids, and they are quite often in a region which is very mobile, then usually this extension is only going up to the point where a transposon sequence is coming, or something very repetitive sequence. So it's not always going kilobases away from your gene, but sometimes you can get back a bit more of the flanking regions. And the other way when we can also complement each other, and this is what I will ask you to do if you have time during the weekend, is when you do the de novo assembly and you find out that there is something wrong because your file size is too big, because you do the check-em and the check-em gives you a very high contamination rate, and you are starting having suspicions that something is there, an extra genome probably, which can come from just simply because your plate wasn't, or the colony which you picked up from the plate wasn't completely a monoclonal. And in this case, the best way to really assemble your genome of interest is to go back to your raw data and filter out all those reads that are not belonging to your genome of interest. So of course there are a few problems here, because what is happening if you don't know what is your genome? If you are sequencing something new and you have no idea what you will get back, so you cannot really use a reference sequence to fish out those reads that are aligning to the reference sequence and then just do the realignment or do a de novo assembly using those reads, because you don't know what is the expected reference genome. And actually, even if you know, so for example, you are looking for an E. coli, but you find out that the E. coli you cultured is mixed with something else. It's maybe not the best idea to use an E. coli reference genome, because as I said before, if you use a reference genome realignment, you will miss those regions that are not in your reference sequence. So if your reference sequence is not containing, for example, any plasmids, but your E. coli actually contains plasmids, then those plasmids won't be assembled later. So the best way, if you do this second approach, is that you try to find out what is the contaminant, what is the unwanted bacterial species or unwanted genome there, and try to find a very good reference genome for that one. Even if it's not known, but even if you can find a metagenomics assembly somewhere, try to find something which is matching well, and then use that reference genome to align your raw reads. And if they are aligning, exclude them. So what you actually do here is that it's like an affinity chromatography. Your affinity beats are your reference genome, and you are not keeping the things which are hybridizing or attaching to the beats, but you are keeping the flow through, and you are working with that data forward. Okay, That's all my slides, so that wasn't too much. If you have any questions, just feel free to ask. Of course, also in the chat, which I can't see at the moment. Now I can see. Okay, so have you finished with your lunch, Jenny?
Yes.
Okay, so you are here with me now. Right. Let's log into the server. I updated my Mac this morning, and I think there was some kind of update which also had an effect on the key chain. So even if you have your, for example, your SSH connection saved in the CyberDuck, maybe it will lose the settings. So maybe you have to set up again. I also lost my private key attachment, so I had to give the location of the private key. So, but probably you don't have to. If you do it like this, as I'm doing now, so if you're using the -I, and then it's working. Of course, not with the Ubuntu user and not with my key, but with the bioinf user.
Sorry, I'm at the moment in the step that I'm trying to find out the program I have to open from my Mac to get the
terminal.
So it's the
terminal.
It's
the terminal. It's in the
yeah. I found the terminal. Yes.
Other and terminal. And you should have the private key somewhere saved.
Is that the one that you sent the new one?
Yeah, the new one.
Just a while ago. Okay, I just copied the new 2 files you sent to my desktop in my computer, so I should have those there.
Yeah, and you have to use the bioinf user, bioinf at 128.232.226.61.
Sorry.
So this one here.
Okay, so I write that one.
Just not with the Ubuntu user, but with the bioinf.
So I'm sorry, I'm very slow at this. So what is the user? Bioinf.
Yeah, it's like this here.
Bioinf. Yes. And then I'm trying to find the. And now I accidentally pressed enter, and I'm somewhere in weird place that if I press the wrong button, and then if I try to write something, it's not the normal place.
Just
in my console.
Push the Control-C.
Okay. Yes.
And you will get back your, and if you just go back up with your arrow, then you will have your last command.
So now I'm so sorry. I'm very. Do you have? Okay.
So -E, and then yeah. So you have to. I think you have to say something like this: SSH bioinf at, and the IP, which is this, and then desktop / I think it was bioinf something. I don't remember what was the key. Let me just try to log in with my, because I also have it on my desktop.
Is it a bioinf key
.pem, I think, yeah.
It's what is it?
Oh yeah, maybe it's
underline. Yes, I have now the 2 files I got today were bioinf key pucci 2 pp.
Not the putty one.
But the p.pem one.
Yes.
Okay.
Yeah, something like this.
Key 2 dot.
It's on my shared desktop as well now.
Yeah, sorry, I had a. Yeah, but it was key/2, I think, because it was the another one, or at least the one file you saved. It had the.
Okay, maybe you saved it like that.
Okay, so then I just try to press enter after this.
Or maybe you have to say yes if you haven't logged in yet.
Terminal would like to access files in your desktop folder. Yeah.
For that, you have to say allow. Yes.
Yes, I did. Are you sure you want to continue connecting? Yes.
Yes.
Is it yes? Yes.
You have to say yes, I think.
Yes. Why isn't enough? Okay, now I'm.
You have already.
Denied.
Hmm?
Permission denied. Public key.
Okay, then go try with the one which is not the 2.
Sorry?
Try the same command. So go
Yeah.
up with your key and try with the PEM key that is not just a simple bioinf underline key.pem. Maybe.
But I don't think I. Okay, let's try. I
There is
don't have
something
that
wrong.
kind. I don't have that file because I only have the key2.pem file in my desktop at the moment.
And this is the one you saved from my second email.
Yeah, I just took it from the. Okay. As I told you, I'm very slow at this.
Did you do it with pushing the tab?
So there is. Sorry?
Because maybe it's because.
There is warning unprotected private key file.
That's the problem. Okay. So what you have to do is that you have to say chmod 600 / wave/desktop / bioinf. So this, of course, with your number 2 somewhere there.
Yes.
Because when you save it from your email, then the permission pattern is not perfect.
Okay, I did that.
And now you have to try again with the SSH command.
Okay.
This one.
Yeah. I think I'm in base bioinf course. So I think.
So you are looking, you saw these system messages as well, and
Yes.
Perfect. Okay, so you are in. Right. So the first thing I will ask you to do is we will download Bowtie and we will install Bowtie for all of us. So go to the Google and Google for Bowtie 2. And for me, it is the very first hit from.
Yes.
So this is the fast and sensitive read alignment. And from here, you can see here on the right-hand side that install with Bioconda.
Yes. So I
just click on that one. And I think this is only what we have to do. So just highlight this thing, Command C.
Okay.
And go back to your terminal and push Command V.
Okay.
So
and then enter.
Conda install Bowtie 2. Yeah, let's hope.
Okay, I did it.
We will see if the repositories are okay.
Something is happening?
No, it's not. For you, not for me. Did you get the same thing as me?
Current channel. Yeah, I think.
Okay.
I think it looks the same. Packages not found error. The following packages are not available from current channels.
Yeah, so we also have to do this. We have to set up the channels. So if you go back to the installation on the website, there is a setup channels link here.
Yeah.
Means that these channels are not added. We used these channels before, but they are not added to the main channel list. These are all different repositories. So the reason why we weren't able to install Bowtie for the first time is because it's not in the default Anaconda repository. So we have to add these repositories. So just simply copy the first one.
Wait, I'm.
Conda config -- add channel.
I can't find those Okay.
Here you have the setup channels link.
Okay. Yes, I didn't press that.
Okay.
Yeah.
And just copy the very first one, conda config -- add channels and defaults. This one?
The 2 setup channels. Yeah, those 3. Sorry. So okay. The first line.
It's okay. Yeah, just the first line, and then we will just rewrite the next 2. We don't really have to copy and paste.
Okay, I just run it or press it.
Yeah.
Warning, defaults already in channels list, moving to the top.
Yeah, I have the same thing. Now delete the defaults and say bioconda. This wasn't. That was the problem, that bioconda wasn't added. And also.
So do I need to do the same?
Yes, because we have different users. So I'm setting up my environment, and you are setting up yours, which is the bioinfuser, which everyone uses the bioinfuser. So you are setting up the environment for all of you. The reason why I'm doing separately, because then I can see the same kind of problems as you have. So, and I want someone. Okay, and the next one is this conda -forge, what we also have to add, and this wasn't added as well. Okay. So if all these 3 are done, then we can go back to our previous command and try to install Bowtie 2 again with conda install Bowtie 2.
Okay. Did it? Collecting package metadata.
It's like mine.
Yes.
We have to wait a little bit, but it's usually not too much, so it won't be that long.
I just have the unprofessional white background instead of black.
Yeah, I think it's it's on the part1 level. You cannot use the black background. I'm not sure that the black background with white characters are better for the eye. I don't know. Okay, so for me, it's already
the dark side.
Yeah. Okay. Yes. Sorry, I'm the dark side. Yeah. Okay. Let's change it to white. So if you have the same thing as I have,
yes.
So that asking for new package, just push enter, because as soon as you see that the yes is in brackets, that means that if you don't write in anything, just push enter, and it will be the default will be used, and it's done.
Oh.
That's all about installing Bowtie 2, one of the programs that you will use probably the most in bioinformatics. It's a very good program, actually. I really like it. Okay, so now let me just leave it there for a moment, and I will show you a few things.
So do I need to do these or?
No, not yet.
Okay.
I will tell you. I just want to show you some very basic things which we will need when we will run Bowtie. So if you just simply say Bowtie 2, then you will get back this quite extensive help. But if you scroll up to the top, you will see that the basic usage is that you say Bowtie 2, and then you can give a bunch of options. Then you have to give the reference genome. I will tell you what does this mean, this BT2-IDX. But this is actually where you give the reference genome you want to align to. And then you will say -1 and -2, which is actually the two read pairs from your next generation sequencing data. I think, yeah, you can also use -u. So if you do the same kind of pre-processing as we did for the de novo genome assembly, at the end, we had 3 different files. We had the unmerged 1, the unmerged 2, and the merged read pairs. So in this case, the unmerged 1 goes here, unmerged 2 goes as the -2, and all the extended or merged reads will go here as -u as unpaired reads. Also, sometimes you get back your sequencing results in an interleaved FASTQ file. So if you have that one, the interleaved means that both read pairs are in the same file. So instead of having 2 FASTQ files, you only have 1. And the program knows that the first record is the pair of the second, and the third record is the pair of the fourth, and so on. So that's what the interleaved means. So if you have that kind of data, you have to use the --interleaved and then give the file name. And then you can give a BEM file if you want. I think you can also use a BEM file for aligning, but actually you will get back a SEM file as the output. And I will tell you everything about what is SEM and what is BEM. This is the whole topic of today, of today's meeting. So you will see all of these things soon. The options you may consider using, there are a lot. And of course, it's always good to understand these, but it's not extremely important for the very first time. So I think the options I usually use this is one of the interesting parts here. So there are presets how the alignment goes. There are very fast, sensitive, and very sensitive. And actually what you see here is that the very fast is a short option, and you don't have to write in this -D5, -R1, -N0, -R22, blah, 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 blah. But you can actually find out what is -D, what is -R, what is -N later. The main differences here are actually the size of the seed, what it uses, I think. Yeah, I will tell you what is the seed soon. And also the number of mismatches it is handling. And there is another thing which is very important to understand here. The end-to-end and the local alignment. The end-to-end alignment means that both I will try to align the whole sequence read, if it's 150 bases, the whole 150 bases based on the parameters you give. It tries to align the whole read to your reference sequence, while the local is only trying to align a piece of it. So if somehow, for example, your reads are hybrid reads, or your reference sequence, for example, fragmented, so you can imagine that if you have your reference sequences in multiple contexts, and you have a sequence read which would align to the end of the contig, but also a little bit going beyond the contig, then it wouldn't be able to align with the end-to-end alignment. But if you are doing with the local alignment, it will be aligned. That piece will be aligned. That is aligning nicely, and then the remaining part will be just cropped or chopped off. So it depends on your goals, of course, and also your raw data, your reference sequence. But in most of the cases, I'm using the end-to-end alignment, and in most of the cases, the very fast or the minus fast is more than enough. Of course, as the name says, the fast will be much faster than the very sensitive. So if time is important, then you should do a minus fast. Of course, today, because we want to do everything together and do it within our time, we will do something like the fast or very fast. These are, again, very interesting things, but I don't want to speak about these, only about the length of the seed. So all of these alignment programs, even if you use BWA or you use Bowtie, these are the two most commonly used aligners. What they do is that they take a small piece from your short read, which is in default, it's 22 bases. I think the program is taking like multiple short 22 bases fragments and looking for perfect match for these 22 nucleotides. So the reason behind this is because it's way much easier and way much faster to find perfect match. And if there is a perfect match for the 22 nucleotide piece of your read, then it will start extending and do the alignment both ways from that piece of your read. So if it's in the middle of your short sequence read, so it's in the middle of your 150 base pairs, for example, then it will start and start checking if the neighbor nucleotides within the reference sequence are matching, or how much they are matching with your neighbor nucleotides on your short read. And of course, this is where all these different settings comes in, that, okay, how many mismatches, it's like maximum number of mismatches in seed alignment, which can be 0 or 1, but it's usually 0. So there shouldn't be any mismatches. But also during the extension step, how many mismatches are tolerated or permitted, like here. So these are the fine settings you can fine tune your alignment. But this is a very important thing to know, that somehow a little piece of your short reads has to completely match the reference genome somewhere. This is just one thing which you should know. And I think what is interesting is again the output. So this is only interesting because I told you that sometimes you can use the realignment to filter out reads. So if you're looking at this one, for example, if you use the minus AL minus cons, or it's actually conc because it's the concordant alignment abbreviated. So instead of, or not, the bow tie will not just do the alignment and outputs the alignment file, but it also put all the reads that were aligning to FASTQ files. So this is the way how you can filter out your short reads that you want to use later on, for example, for de novo sequencing by using a reference genome, or the same kind of thing, but the opposite is using the minus U and conc, which means that you are putting all those reads into a new FASTQ file that are not aligning. That was the approach I told you should use, and this is what you may use if you want to assemble the genome that I will give you for the weekend. So the raw data, in that raw data you will have 2 different genomes, or 3. I think I will just give you the one with 2 different genomes, and you can use one of the genomes to do a bow tie alignment and extract all the reads that are not aligning to that genome, because you actually want to de novo assemble the other genome. So then you will use these options, but I will give you some help for doing those. So don't worry if you don't memorize everything at the moment. Just know that there are these special commands as well with bowtie2. And of course, as always, you can increase the performance with -p or minus threads. So again, here you can see the number 1 in brackets, which means that in default, bowtie is only using 1 CPU core, but actually multiple alignment is very easy to do in parallel, so it's scaling very well, which means that if you are using 4 times more cores, probably the time will be 4 times less. And I think this is mainly the things what you have to know. And before we do our first alignment, and this can be done by everyone. So the first thing what we have to do, and because we only have, I didn't want to replicate the reference genome. We can use the same reference genome, all of us. Let me just do the reference genome indexing. So as in most of the programs, if you do BLAST in console mode, you also have to transform your database a little bit. So BLAST cannot use a simple multi-FASTA file as the database. You have to create a BLAST database. In this case, we have to create a bowtie database, which we will do with bowtie2db as far as bowtie2 build command. So if you look at the usage, what you have to do is just say bowtie2 build, and you will put here your reference FASTA file, and you will put here the prefix for your database. So I already downloaded a Staphylococcus aureus reference genome, because we will work with the same raw data as we worked before. So if you go to the Scratch folder, there is now a folder called data. Oh, and let me just make it accessible for everyone. And if you look into, there is only one file at the moment, which is actually a Staph aureus genome. I think it's a complete genome, so it's not fragmented. We can also always check how many contigs we have. If you remember, there is a grep command, grep -c, and this thing. So you are counting how many lines have this symbol in it. If it's only one contig, if the genome is complete, then you will always have one line, and this is what we have now. So this is a complete genome with only one FASTA record. If you want to see what is the name of this FASTA record, or what is the identification, then just use the head command. You can always see the first line, which says it is the Staph aureus strain SA40TW, which is maybe not the most the closest genome to what we have, but it's good enough, I think, for our purposes. So what I have to do, and this is also something you should do if you do your own data sets, but in this case, it's enough if only I do this, is the bowtie2 build. So make the database. I will give input FASTA file, and I will give a name for the database. I don't want to give this very long one, because we have to give that database name. So let's just call it as the our ref. And if I do this, yeah, I remember that if I push enter now, then my screen will be full of a lot of unuseful information. So you can always use the -q. So if I scroll up to the bowtie2 build command, there is a -q option, which means that it will be quiet. This is actually not true. I think this is a bug in the program. So the -q means that it will be quiet and it won't be verbose. The verbose means that it will give you a lot of information. But if you say -q, there may be only a few lines. Okay, nothing happened. So if I'm saying ls minus l now, or even minus lh, so as you can see, we have the original FOSTA file here with about 2.7 megabytes, which means that it's about 2.7 megabases. And then we have different index files and database files. You don't really have to know what are these. I don't know what are these, but probably they are, I think maybe there are like small pieces or small k-mers, chunks from the reference genome and their positions. So, but anyway, it's not important for us. We just have to use this thing here as the prefix in our bowtie2 run. So if you go back to your own folder now, then I don't know if you have any folder, Jenny, already. You don't.
No, I don't.
Okay, so if you are in the Scratch directory, if you just simply say cd spare scratch, and you can make your own directory, mkdir, and then the name you want. So
I think Jenny is not taken, so and
let's see. Oh, it's there actually, or you just
I just did it.
Okay.
I was fast.
Yeah. Okay, and because you didn't do the things with us, I will give you those files, which we will use today.
Thank you.
So as you can see, I have the deduplicated FOSQ files here, but I also have, these are the files which came out from the flash merging step, the not combined 1 and 2, and also the extended frags. Well, let's work with these with these ones. So I will just copy. Okay, these are not small, so maybe it will take a few seconds.
Yeah, now I have 3 files in my folder.
Yeah, but I think it is copying, so.
Okay.
Okay, it's done. Let me just check if you will be able to use them, because I copied them as a root, and of course it's having the root user, but it's not a problem because you can read it, and you only have to read these files. You don't have to write them, so it will be perfectly okay. When you try to delete these files from your folder, you won't be able to, but don't worry, I will do that.
Okay.
Or actually what I can do, just simply almost. Okay, so now you have your control over the files. Okay, so what we have to do, if you are in your own folder now,
Yes.
so and everyone else can do this as well, we will just execute this in one core by using one core, so I don't think that they will slow down each other too much. So as you remember, if you just say bowtie2, then you can see the usage here. So we have to give the -x and then the prefix. So bowtie2 -x and the path to the reference sequence database, which is /data /st ref without the dot, just this thing. So this is pointing to the already converted reference data, reference sequence. And this is the prefix of the bowtie database. And then we have to say -1, which will give, where we have to give the mate1, the FASTQ file, which is, I think, out.notcombined underline 1.fastq, and then -2 out not combined 2 fastq, and then the unpaired reads, which is, I think, -U. Yes, -U, the out.extended fastq. So if you haven't been here when we did this, Jenny, the extended fastq file is those reads that were overlapping in the middle. So when you are doing, the parent sequencing, sometimes because of your library size, the two reads can actually sequence the same thing in the middle, because the sequencing is going to from the two ends into the middle, and we used a program that is detecting these overlaps and make one read from the two by merging them. And before you press enter, no, don't press, nobody, don't press enter, because the problem, if you press enter here, or if you pressed already, just press Ctrl+C. Because first of all, we also have to give, I think, this one of the presets. So let's just give a minus very minus fast. Is it minus or underline? Yes, so it's minus very minus fast. And don't press the enter yet. If you do this, then what is happening is that bowtie2 will give you the alignment file back, but it will give you to your screen. And you don't really want to have that SEM file with millions of lines in your screen. So instead of receiving the results on our screen, we will put it into a file. Okay, so just put this arrow after your command and say output.sam. And again, you don't have to give the SAM or SEM extension, but we know that it's a SEM file, and it's always good to know that what we will have is a SEM file, because then it will give us the clue what to do with it. And just push enter, and then we can all see how fast is this with one CPU core, and with all of us running the same thing at the same time. Let me just check how the server is doing. It's actually not too easy to log in. Yeah, but there are nice alignments are running. Mine as well. And also, if I check my folder, so I am running the script in this window now, and as you may also run. And if I'm saying ls-ltr, which means that the files are ordered by their creation date. So the last file is always the one which is the most recently created one. As you can see, the SEM file is already quite big, and it's going up. It has to be at least, I think, as big as these two together. So there will be, I think, a few minutes while it is running, because actually the SEM file is containing almost everything, even a bit more. than the FASTQ files. We will see soon. So yeah, I think it has to go up to 1.5 gigabytes as well, at least, I think. Yeah, so while it is running, I can speak to you about the BWA. I don't know if we will use BWA today, maybe not. When I first did my very first whole genome realignment, I used BWA, and somehow I think the eukaryotic whole genome realignment pipelines are, for some reason, they are suggesting to use BWA and not bowtie. And in the last few days, I tried to find out what is exactly the big difference between BWA and bowtie, but I didn't really find anything convincing. So I think you can just use any of those you want. As far as I see, the bowtie is a bit faster, but I'm not sure. I'm sure that the BWA is also quite frequently refreshed and new. But actually, if you look at my screen now, the usage of BWA is very similar again. So you have to index your reference sequence. This is how you use the BWA. So it's the same kind of process what we did with the bowtie build. So it will do some kind of indexing on the reference sequence. The index file will be there. Probably it's just another file with the same file name but a different extension as your reference sequence. And then you do either this thing when you only have one FASTQ file with your reads. So say BWA mem. The mem sub command is like a sub process within the BWA, and this is the one which is used for alignment of short reads to large reference sequences. So BWA mem, and then you are giving the reference, which is of course already indexed, and then your reads file. If you only have one FASTQ file, if you have multiple FASTQ files, then you are just doing this thing 1 and 2, and the same way, instead of putting the alignment results to your screen, you are just putting it into a file. So very, very similar. I don't think that it's really worth to spend a lot of time with this one as well. But again, you can use multiple threads if you want with the -t command, and you can also fine-tune the alignment with the gap and match score settings. Let's see how the running is going. Oh, I'm already up almost 1 gigabytes already, so I think it's going well. Let me just grab some water while it is running. If there's any question, then please don't hesitate to ask.
Lajos, I have a question about the non-combined sequence. So usually is if you have a sequence, have 2 pair ends, and then is you use the FASTQ file, use the paired end FASTQ file to convert to SAM file. But usually you lost the single end sequence. Is this a matter or not?
In our case, we are using all three files at the moment. So we are using the not combined, and we are also using the extended here. So that's why if you look at my screen, we are having the -u, and where there we have the extended fracs.fosq. So these are the single end reads. So of course, the not combined 1 and not combined 2 has to be exactly the same length. So you have exactly the same amount of FOSQ records in 1 and 2 because they're each other's pairs.
All right, I got it. And another question about the procedure of how to deal with these files. Is there like any how to say that? Is usually you get the paired end and you merge that, and then is align that is with the reference sequence. So how about if you do the other way round, like you do the alignment and then is merge the two pair end sequence?
Yeah, that's why so that you don't really have to do the merge. Right? For the realignment, it's not as important to do because actually you are just aligning them to a reference sequence. So it's completely okay if you do, for example, this alignment, what we are doing with not the flash output, but those files which we used before we ran the flash. So if I'm going to my folder, so I have, we had the original raw data, which is in our raw data folder. So A, you can use these FOSQ files. And this is just the pair. B, you can use the deduplicated ones. The reason why it is better to use the deduplicated ones because you are just saving some time because all the duplicated duplications were removed. So instead of aligning 100 million, you're just aligning 90 million. So you are saving like 10% of your time. And if you just use these ones, then you only have to use the -1 and -2 options with both I2 because you don't have any unpaired reads yet. I think the flash merging is way much more important if you are doing the de novo assembly, because if you have a chance to merge reads, then and you can do it, and you can, let's say, merge 20-30% of your reads, then you have a chance to have much longer reads, and much longer reads can cover more sequence. So you will have a chance to build better context because you have longer reads. But it's perfectly okay not to merge any of these reads before, because it will be anyway, it will show up in your alignment file that they are overlapping.
Okay, yeah. Thank you. Yeah.
Mm-hmm.
I think mine is done.
Oh yeah, I have 2 GB now, so I think it's very close as well. What's the size of your SEM file if you say the same kind of ls-lt-rh like me?
Can you show the?
This LTRH.
Yeah. And output sum file, it's 2.0 G.
Okay, so it's the same as. Oh, okay, yeah, mine is finished as well. Okay, so first of all, what you can see here, you will have a kind of report of your alignment. This is just giving you the number of reads that were paired. So this is coming from the merge, of course. And the most important thing is the alignment rate, which is the very bottom here, which is, I think, quite good. So 91% of the alignment rate was, again, you can start think about why it's not 100%. I guess, I don't know if, I mean, Ibrahim, if you know anything about these isolates, I can imagine that they have plasmids in it. I know that they have AMR genes. I don't know how many of them are on plasmids, but I can imagine that there are plasmids in these.
Yeah, I think that the difference would be the AMR genes.
Yeah, I mean, not just the genes, but probably plasmids, so mobile genetic elements. So again, if you are really sure about your purification, DNA purification and colony selection and DNA extraction, and you think that there should be only one genome in your sequencing sample, then if you see something like this, that means that you have some extra DNA there. So this is what I told you, that probably we won't see any AMR genes in our realignment file. I don't know if the MAC-A is there in this reference genome or not, but that means that you are losing important information. But again, we are not aiming for finding everything now. That's not our final aim in this case with realignment.
Lajos?
Yes?
Because then you get like an output where you have all the non-aligned sequences. So if you're looking for those particular genes, would you then look in that folder directly, right? Because you assume it's a mobile.
Exactly. So what you can do, and don't run this, just memorize or just come back when you want to do something like this, or when you want to repeat this, is before you close your command line, your command, you can say minus un minus cons, right. So this one here, and give some kind of file name. This will be like non-aligned reads or something. There will be two files actually. So there will be a non-aligned reads 1, non-aligned reads 2, because right. The pair and reads will be put into two files. Maybe there will be actually.
Oh, sorry. What happened with the extended?
Yeah, I think there will be 3 files actually. Okay. I haven't done it with, but yeah, I think there will be 3 files. We can just try later on, or you can just try if you log in back and see. And actually, you can use this non-aligned reads to do a very simple de novo assembly. Just these ones, just going through the whole de novo assembly, and probably you will assemble a few really nice contigs.
And then I have another question, because then I'm assuming this is like very high percentage, basically because we're looking at one particular genome, right? But if you're looking at metagenomes, that then that percentage should decrease, or do you use still bow tie, or there's another?
I mean, I wouldn't use any kind of realignment technique in metagenomics. So the only reason why you should use realignment in metagenomics, if you really want to find your genome of interest in it. So if you know that in your sample there has to be an E. coli, then you can use an E. coli reference genome and find all those reads that are aligning to the E. coli, and then build up your E. coli and try to find the MLST, for example, the sequence type of your E. coli within that sample. But that's only working for those genomes that you know. But even if the best known human complex microbiomes, like the fecal microbiota, 80-90% of the species are not known yet. So we are just having too much unknowns there. I have that figure somewhere. If you have just 1 second, I can show it to you. when we did I show this in on the course, the E. coli realignment? Yeah, this was it. So if you look at this figure, this is actually what you said I did. This was done by Botai2.
Mm-hmm.
I had 100 fecal samples from pig farms. So these are pig fecal samples, and I just checked the presence of E. coli in them. And I think the highest level was, the alignment rate was like 1.5 or 2%. Mm-hmm. So, you know, that means that, of course, you can fish out your genome if you want. But, and the E. coli was there, even in these ones, there were some E. coli genome. It was very, very low amount. So the bug was very low abundant.
Yeah.
But yeah, so the question, the answer is yes, if you know what you look for. Otherwise, you shouldn't.
Right.
And yes, you can say something from the alignment rate, because I can, if you know that you are just looking at bacteria, for example, if you do a bacterial pellet from your complex microbial sample, and you know that there are only bacteria in this, and you see that your overall alignment rate was 2%, then you can see that in this sample, my bug was there in about 2% abundance, because, you know, prokaryotic genomes are usually quite close to each other.
Yeah.
There are sometimes like a two-fold difference, but not really often. So it's, you know, they are between 2 and 5 megabases usually. So if your alignment rate is 2%, that means that probably your genome was represented in about 2% of all the DNA.
Okay.
So it is okay, but in this case, if we really want to find out what is in that almost 9% of non-aligned reads, then you can do this kind of extra option and command, and then by having these non-aligned reads, you can do a de novo assembly, or you can check them by Kraken, or you can check them by Ariba to see if there are interesting genes inside. Of course, Ariba is only checking for those what you look for, not for everything. So it's maybe better to do a de novo de novo assembly, sorry. Okay, let's look into the SAM file, because that's very interesting actually. So this is how the SAM file looks like. There are some headers, which is not extremely important for us at the moment, but here you can see what was the reference sequence and what was your command, line command, how you generated this SAM file. And again, my window is not wide enough, but actually this is one line. So instead of having the 4 lines, it actually looks like 4 lines, but it's only 1 line in reality. As you know, in FASTQ files, one record is in 4 lines, as we spoke before. The first one is the ID, second sequence, the third one is nothing, and the fourth one is the quality of the sequence. In the SAM file, the things are everything is in 1 line. Actually, the very first one is not aligned. That's why we have a lot of zeros here. But if you look at this one, and this is ending here. So if you look at this line, what you can see is that you have the read identifier here. This 99. I have to remember this. What is this number? I think it's the length. Yeah. So there were some, I think, some shorter ones. I think this is the length. No, it's not the length. It's something else, but it's not that important now. Then there is the sequence here. The position is also here. So there's a from and to. No, I think it's just the from. here. Okay, let me just. It's much easier to give you. Okay, so there is a flag there, which is here. So the 90, was it 90? 99. And it's probably 64 plus something, which we, okay. Let me just skip this. The most important things are the reference sequence name, which is always the same because we only used one reference sequence and only one FOSTA record. So this is the reference sequence name here. And then we have the position within the reference sequence. So this read is aligning to this position on the reference sequence. Then we have the mapping quality, a cigar string. This is a very interesting thing. So this is only tell us that actually the read is aligning with 146 nucleotides, which is completely matching. I don't know if I will be able to find something which is not completely matching, because we have all the non-aligning ones here. It's not the best way maybe to find something. Oh yeah, here. Sorry for just scrolling and scrolling down, but for example, what you can see here, that means that within the whole sequence from here up to here, the first 45 nucleotide is matching. Then there is one insertion compared to the reference sequence, and then there is 104 nucleotides that is matching again perfectly. So this is a kind of estimate on what you want, what you have as an alignment. But of course, you will never see this SEM file, but it's always good to know what is in the SEM file, and also good to understand. And then there are some other strings, but these are actually not extremely important at the moment. The more important ones are the sequence and the quality. And again, you won't do anything with the SEM file. If you look at the size, there was that few header lines, but it's actually a huge file. So it's, as you can see, it's 4.5 million lines. So there are 4.5 million -3 header lines sequence aligned here. Of course, the sequences that are not aligned, as you saw, they are also in the SEM file. So you can actually extract the reads from the SEM file too. So if you make your own script and extract all the reads that are having 0 here, which means that they are not aligned because they are having 0 at the position of the start, and also an asterisk in the position of the reference sequence. So this is the third column. Instead of the reference sequence ID, there's an asterisk. And in the fourth column, instead of having a position, it is 0. And if you extract all the lines with this, then you will get back the sequences that you can, again, use to check if there is something interesting in those sequences. Okay, so what we will do next is we will try to visualize this. You can visualize a SEM file. You can also store SEM file, but SEM files are big, and it's not the best to store them in this way. But actually, it's always good to store your alignments because this is usually the most time-consuming step in a whole genome realignment, especially if you are doing eukaryotic genome whole genome sequencing. So if you are doing a human whole genome sequencing, that this realignment phase can last for days. So you don't want to redo your realignment, which means that you want to save usually one of your files. And it's often not good to do with the SEM file, but it's better to do with a BEM file. So we will convert our SEM file to BEM file. The first thing what we have to do, and now I'm asking your help, Jenny, again. So we have to install SEM tools. That's again a program that you will probably use a lot. SEM tools. Just look for SEM tools in Google, and it's probably the very first hit. No, yeah, the download link is here. I'm just checking if there's any conda. I think there should be a conda install for SEM tools. Let me check. If not, then we will install in the old way. Oh, yeah. I think we can do just simply this. Conda packages SEM tools. Maybe this is not the most recent one, but actually it's 1.1 there as well on the SEM tools website. So let's go back to your home directory, Jenny. So just say cd, and this will give put you back in your home directory, which is if you say pwd, it's your /home/bioinf, and here just simply copy and paste this conda install -c SEM tools. -c bioconda SEM tools, sorry. So it's using the bioconda repository for installing the SEM tools. This is a bit easier way than downloading and then compiling, and I can actually also install SEM tools centrally. So if this wouldn't be like a test or learning server, but it would be my own like in-house server, and I know if I know that there will be many people using SEM tools, then probably I should install SEM tools as a super user and make it available for everyone. But I think in this case it's not bad to practice. I really hope that everything will be installed, also the htslib. If not, then we can just install later. This is again very fast because it's just 1 megabyte. So, and we have the SEM tools, so I think it will be okay. Did you finish already?
Yes.
Right. Okay, so we have to do two things before we can open this file and look inside. So the first thing we have to, now actually we have to do three things. The first thing what we have to do, that we have to sort the SEM file. It's not, I think it is not obligatory, but the reason why we are sorting the SEM file is because if it's sorted, the sorting means that the records will be in the order of their position. It's not now, it's not in that order. Now it is in the order, I think, how it was read in from the FASTQ files.
Question: when you wrote the SAM, and I did the same, I got that command SAM not found, but there are 18 similar ones, so I don't get the same.
Because I pushed the tab.
Ah.
And if you push the tab, then if you just push the tab after SAM, nothing happens. If you push the tab twice, then it will give you the options.
Ah, okay.
So there is a SAM2 vcf.pl, there's a SAM2s, and there's a SAM2s.pl. Which I don't know what is SAM2s.pl, but actually we are only using the SAM tools. So as soon as you say SAM, then it will be copied to SAM tools, and this is what we will use.
Okay.
Okay, so back to the sorting. The reason of why we want to sort the file is that because we want to compress later, and that's where you will use the BEM file conversion. and the BEM file conversion and compression is much better if the SEM file is sorted. So what we are doing is that we will sort the SEM file, and actually I will go back to here and look at the comment here. So actually the SEM tools has a very good online help. And if you look at the SEM tools sort, then you can see what you have to give. So what you have to give is actually just an output file name, and which can be a BEM file. I don't think if you give a SEM file, it will be a SEM file. If you give a BEM file, it will be a BEM file. So we will give a SEM file, and we will output a SEM file as well. So if we go back to our folder, cd per scratch per virus for me, and I say SEM tools, and this can be done by everyone. SEM tools sort, especially if you have your alignment file, output.sem, but before this will go to the end of the command. So if you see the input file is going at the very end. If it's a SEM file, BEM file, or a CREM file, we can also use the -n. You shouldn't use the -n. -n is sorting the reads by their names. There are a few cases when you should do this. For example, I'm doing it for the high C metagenomics, but in many cases you don't want to sort them by the names. You want to sort them by their position on the reference sequence. And I think the threads should be set as well. So the -@ sign. If you look at this, then you can give a number there for using more than one CPU cores. Again, we are only using one here because then we can do it in parallel. So the default is one single thread. Okay, so we are just giving a -O, and it will say output sorted dot SEM. So that will be the file name for the output, and I think this is it, but let me just check. Yeah, I think it is running well. So you can also start running it. And it is running for me already. Let me just go back if you weren't. So SEM tools sort -O output underline sorted dot SEM, and then
I got it.
And we can also check the file sizes. And it's not there yet, so it actually has to be the same at least, but actually it's doing a BEM file. So maybe we will save the time for doing the BEM file. We'll see. No, I think it will extract the SEM. Oh yeah, okay. But it's actually very fast. It's almost the half is done. The file size should be the same. I don't know if the unaligned ones are in. I can imagine that there is a command to exclude the ones that are not aligned. Maybe not. Maybe not. Oh, it's done for me.
And I think it's done for me also.
Yeah, it's almost exactly the same size. So there are 2 extra bytes written probably into the header. If we look at the output sorted now, this is what we wanted to see. So I think, yeah, I think maybe this one, that it is sorted by the coordinate, maybe this one went in as an extra information. But as you can see now, the position of the starting position. Was it the fourth column? Is it here? Yeah, the position is the fourth column. So as you can see, the first records are all starting at position 1, and then 2 and 3 and 4 and so on. So now it is nicely sorted. And the next thing what we want to do is to convert this to a BEM file. And this is what we will do with the same kind of tool set, the SAM tools, but instead of using the sort function, we will use the SAM tools view. The SAM tools view can read in the SAM file and then make another SAM file. So actually the SAM tools view is also good if you want to extract a sub section of your sequence, but it's also good for converting the SAM file to BEM file. The only thing we have to do is that we have to say -b, and instead of then putting it into a SAM file, it will put it into the BEM file. I think we also can also set up the compression rate, and this is what can be interesting actually. So if you say -u, then the BEM file will be uncompressed. The BEM file is storing the same thing as the SAM file, but it is storing it in a binary format, so it won't be readable for you anymore. You cannot just look into the BEM file with the less command and see the sequences inside. So it will be translated to a binary file, which means that it will be already much smaller than the SAM file, but you can also compress it internally. If you use higher level of compression, then the file will be smaller, but the whole compression will take longer, will take more time. So I think we shouldn't do anything with the default settings. I think the default compression rate is somewhere in the middle. It's not too fast and it's not extremely compressed. So what we only have to do is we have to say SAMTools view -b and then the output file. No, the output file. Maybe with the -o we can give the output file name if we want. Yes, otherwise it is going to the standard output. So what we have to do next is say SAMTools view -b. So we want the output as a BEM file, -o output dot underline sorted dot BEM. So this will be the output file name, and then we are giving the input file name, which is the output underline sorted dot SAM. I think this will be quite fast. I hope. Okay, it's not just me who is using the SAM tools. I think you can, again, you can do it with multiple cores here. So as soon as you are doing it yourself, maybe it's the -@ sign, yeah, the same. The SAM tools is always using this -@ sign. So -@, and you can use 4 on this server. Oh, it's actually, oh, this is interesting. So it, instead of setting the number of cores, it is setting the number of extra threads. So if you are alone in this server and you are running the SAM tools, you can set this to -3 actually, because that means that you are using 3 extra threads as an addition to the main thread, and then it will be altogether 4. is almost done. At the same time, Jenny, please download the, oh, it's done for me.
Yeah.
Please download the Artemis, because the Artemis also includes the BEM view, as far as I remember.
So what do I need to do?
I will show you. Yeah, the BEM view is there. Okay. So what you have to do is Google for Artemis and Sanger. Otherwise, there are a lot of different Artemis things. So
Yes.
if you just Google for Artemis Sanger, then my first hit is this thing. And what you want to do is, of course, accept cookies and downloads up here.
Yes.
It will come down quite quickly. I set downloads. Maybe it's here. Yeah, okay, so the GitHub page link here.
Okay.
And then go down to the, no. Yeah, go down to software availability and go for, no, you have a Mac as well.
Yes.
So it's a very nice tool, and actually you can use it on Unix, Linux, Mac, and Windows as well. You can also install it Bioconda, but we don't want to install this program to the server now. So you want to install this to your own laptop, because this is a graphical user interface program. So you will look the BEM file on your own laptop. Okay, so that just push the Mac OS version and it will go and download. It's I think it's a DMG or PKG file. It's actually DMG. It's maybe 100 megabytes, so there will be a little bit of time. While it is downloading, we already have the BEM file. So now we can see the sizes. So as you can see, we almost saved or reduced to it's like just to 20% of the original SAM file size. So this BEM file is having exactly the same amount of data inside that the SAM file above, but it's like 5 times smaller. So that's why I say that as soon as you finish with your realignment, you can just simply delete your SAM files and store the BEM file. You can always get back your SAM file. So if you do the same thing, what we did with the SAM tools view, if you do this and you say -o output sorted SAM, and you are giving the BEM file as the input, then the whole thing will be converted back. So in this case, I am giving the BEM file as the input, and I am setting the SAM file as the output. Because the SAM tools view, if you look at the SAM tools manual page, the SAM tools view is prints the alignment in the specific input alignment format, and the standard in default, the standard is the SAM format. Okay, maybe you have to give -s, but I don't think so.
So I got the Artemis or downloaded it and clicked it, and now I have this window Artemis tools.
Yeah, I think what you have to do is just if you are, let me just show you, if you are here with this thing,
yeah,
and you have the 4 icon there,
yes,
then just simply drag them and drop into applications.
okay.
All 4 of them, okay? I think you can do it maybe at the same time. I can imagine that you can do. Oh no! Oh, I double click. No, cancel. Whew, okay, I survived. But I think if you just drag and drop to the applications, then
yes,
you will, you can just access from the programs. It will be probably on the last page. So if you have to go to you know how to access this.
Okay, so is it the Artemis, or Because I have Act, Artemis, BAM view, and circular plot.
Yes, we will use the BAM view, but not at the moment.
Okay.
We will have to, oh, we don't have time to use the Artemis, but we will just try to use at least the BAM view. Okay. There is only one thing what we have to do before we will see the BAM file. And this is, we have to index the BAM file. This will be very fast. It's actually just saying CEMTools index and the output sorted.bam. As far as I remember, let me just quickly check, but I think it's only this. Yeah, line sorted BAM, and you can give the output file name, but you don't have to, because it will be there as far as I remember. So simply CEMTools index output sorted.bam.
Okay.
As far as I remember, BAM view needs this index file as well. So if it went well, then you will have this BAI file as well in your folder. So not just the BAM file, but also the bam.bai.
Yes, I have it.
This is the index file. Okay. So what we have to do now is we have to download these files from the server to your own machine. So I don't know if you already set up the CyberDuck. Do you have the CyberDuck?
Probably not.
Okay, then don't do this at the moment. I will send you the link. Just look at what I'm doing.
Okay.
Everyone else who has the file transfer set up, please download the file. It will be a few hundred megabytes, but it's okay. So, and also download the reference genome. I will show you from where. Oh, what do we have? Oh, someone else did the database as well, not just me. It's okay. Okay. So I will just drag these two files and drop to my desktop. It will be a bit more than just 1 minute, so it's not extremely a lot. And I really hope that I will be able to, I used BAM tools for 2 years continuously, but 3 years ago, so forgive me if I won't be perfect in the usage of BAM view, but I guess I know how to use it. The reason why you really want to see this in the graphical user interface as well. Of course, if you are looking for example, SNPs, if you want to check how many SNPs you have or how many differences you have between a reference genome and your own sequencing, you will not sit down in front of the BAM view and go through the whole 2.5 megabases by hand and grab a pen and record all the differences you see. For this, we will use a program, which we will discuss next time, it seems. But if you use a program to check for differences, and you want to go back and check if it's really a difference, if it was a good call, if you see a nice coverage in that position, and you really see that differences in many different reads, for that you have to use the BAM view. For me, it is downloaded to the BAM and the BAI file, and I will also download the genome file. So I go up to the Scratch directory and then go into the data directory and just grab this GCF blah 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 file as well. This is much smaller, just 2.8 megabytes. So you will need these 3 files. And if you open the BAM view, on Windows, maybe it's a bit more challenging to open the BEM view. As far as I remember, you have to have the Java or Java runtime environment as well there. Okay, so this is what you get. Here you have to include the BEM file. And as far as I remember, oh, this is such a nice thing. So if you go on your desktop, if you copy it, of course, you have to allow the BEM view to see your desktop. And I have the output sorted BEM. I really hope that it will recognize the BAI file. If not, then we maybe have to give it as an extra. We will have to maybe rename it. I will see. But anyway, this is the BEM file, which I copied, and I also select the GCA reference sequence file. I have to say all files here, and I have to give this GCF something. Maybe it doesn't allow using the FOSTA file, and then I have to convert it to the FOSTA file. We will see. Oh, it's actually here. Yeah, so as you can see, we have a very nice coverage. With the blue ones, you can see the read pairs. Oh my God, it's not really nice. And with the black ones, you can see the unpaired reads. We can zoom in, and then we can also see the nucleotides probably after a while. Yes. But actually oh my God. Yeah, it is here. I don't really see the reference sequence here yet. I am not sure the reference sequence. You probably have to convert the reference sequence to something else, because in this case we won't see any differences. Yeah, there are some in the very beginning, but I don't think we. No, oh, there are. Okay. Sorry again, just scrolling into the sequence, but this is what you want to see as a difference here. So the blue means that it's completely aligning. Oh my God, it hurts my eyes. This blue color. So the blue means that it is completely matching with the reference sequence. And in this position, there is a SNP. And as you can see, it is there in all of our reads. You can see a few reads here. I don't know if you can see this. I will try to increase the height. Oh, maybe I decreased it. Yeah, so here we have a few reads in the middle, where at the very end, there was probably some missequencing and the end wasn't perfect. But actually, in this case, this position is almost in every other ones are good. And you can also see the mapping quality here. So if you go over, you can see the read identifier, and you can also see the mapping quality, the map queue, and maybe the mapping quality here. Oh, it's actually not so bad. It's just probably because it's at the end. I think it's the mapping quality for all of them. And if you go in, oh, it's probably an insertion. It's not even a, yeah, it's an insertion, sorry. So there is an insertion here compared to the reference sequence. And that's why you have this vertical line here. Let me check if we have. And sometimes it requires quite a lot of memory as well from your laptop if you want to see, especially if you're zooming out and in, as I'm doing. It is just slowed down a little bit now. Okay, I will not try to find anything like this anymore. What I will do is that I will, for the next time anyway, we will do the variation calling. I will go for a certain position and I will show you how to use the BEM view properly. But this is actually what we will need to do. In some ways, we have to do the BEM, the index file, and then you can just copy and view it on your own laptop. But as I said, you never search for variations graphically. You can search for those ones which were found with something else. Right, so I think I will just finish here today. If you have any questions, then please ask. And I will write you an email very soon with all the instructions if you want to practice any of these things, even the de novo assembly or the realignment during the weekend. So I will give you some file locations you can use to do your own assembly. I think we are now at 20% of our storage space, so you can also do some cleanup after. I mean, especially in your own Scratch directory, so you can, for example, delete the SAM files easily because there is no need for any SAM files anymore. So you can just, if you are in your Scratch directory, you can just say rm-f star.sam, which will delete all the SAM files. And for me, this is freeing up 2 times 2 gigabytes. Right, any questions?
I just have a little question. I don't know whether you said it. Oh, maybe I lost that. But what means the in the Artemis, the lines that, or the reads that are in black? Does that say anything, or it's just like?
Yeah, I think in the BEM view, the black lines are the ones which are not paired.
Ah, okay.
Because you also see the pairing sometimes. I think this BEM view is not the one which I used. I will try to find that BEM view. It was much more user-friendly, to be honest. I don't know why it's not working for me nicely at the moment. It's also, I think, just hanging. So I will just quit from it now. But I will show you that kind of way I used to use. I just have to spend probably an hour to set up the BEM view in that way that you really can see a lot of things. For example, you can see which read is paired with which one. I think it's only like setting the view, but I don't want to spend the time with that now because we are already running out of time. But I will show it for next time. And it's also interesting. It will be also interesting next time because what we will do next time is also we will do an automatic variation calling. So instead of looking for SNPs or insertions, we will apply a program that is going through the BEM file, compare it with the reference sequence, and give you a list of the potential variants. Okay, and then if we see something interesting, then we can go after that. And I really hope that the BEM view will be also much nicer than this. I'm stopping the sharing now. If there is no more questions, and then I hope that you learned something today as well.
Yeah, thanks, Lajos.